Hello everybody, my name is Konstantinos Makris. I'm a professor of environmental health at the Cyprus International Institute for Environmental Public Health at the School of Health Sciences within the Cyprus University of Technology, and today I will try within a relatively short time, within 15 minutes or so, to present to you the case for considering seriously the human exposome concept and its applications, and whether the future of public health, the future of biomedical sciences, and the future of the quest of understanding disease and predicting disease is actually here. And to that extent, I would like to start my presentation by providing a short synopsis of what is the human exposome all about and the different definitions that are currently applicable, the components of the human exposome, the methodological framework that packages this concept, the tools that come along with it, and how these tools are actually used and integrated into research, and I would say in some clinical applications. And this is how I'm going to end my presentation. Therefore, since the launch of the Human Genome Project, maybe 20 or 30 years more, including the preparatory activities, much research has been conducted and billions of dollars have actually been spent on better understanding the contributions and the role of genomics in chronic disease processes, including both prevention of chronic diseases and including control. However, this gene-centric approach, because this was the emphasis of the Human Genome Project, eventually yielded limited results and not the big results that initially were anticipated. And as the knowledge accumulates, it becomes more clearly understood that the majority of the human traits are both polygenic and polyexposure based. And talking about polyexposure based, this term refers to the different environmental exposures that people are usually exposed to during their everyday life, and the emphasis that we shall give to that. And the emphasis we shall give to this kind of multiple human exposures comes from the fact that they shape everyday life and they're actually part of the equation. I mean, the main equation of the disease phenotype, for example, is actually equal to the genotype contributions, the effects of the genotype on the phenotype, plus the environmental exposures that humans are daily or systematically being exposed to. Let alone, and let's not forget another model term, this is actually the interactions, the G times E interactions, the interaction between these two main effects that contribute to the phenotype. Therefore, we would like to put in this equation this second part of the model terms that includes the environmental exposures and the interactions of these environmental exposures with the genotype. And if we do this, we are actually becoming more actively involved in the so-called new biology era that follows up the Human Genome Project era. In that, more multi-omics approaches are actually being included in this. And I will explain later in the next slides what are these multi-omics approaches are all about. So a key piece of that puzzle in understanding disease, preventing disease, predicting disease, we believe it's the human exposome concept and its groundbreaking tools, including, of course, its life course framework and the critical windows of susceptibility and vulnerability that come along with it. So if one would holistically characterize this, all these non-genetic environmental components of chronic disease, but not stopping there, but trying to integrate those environmental exposures with the different layers of endogenous response that actually become stimulated by the external environmental, exposures, then this combination, this integration, or better say, working at the interfaces between the human external exposure characteristics and the internal response, biological response, the biological effects, hopefully this integrative approach would allow us to unveil new discoveries in this new biology era that deal with chronic disease, in particular prevention and control applications. So to this extent, the human exposome can be very, very handy. Why? Because heritability, particularly the heritability that is captured by the twin studies or the family-based association studies, appear that this kind of heritability is not solely dependent on genetics, but it's rather controlled by functional interactions between genes, their products, and the environment. So we would really like to consider not only the genetic contributions to the disease prevention and control, but also the shared gene times environment interactions, which usually being ascribed to genotype contributions, and this is often becomes part of the missing heritability that we usually tend to include when we cannot really ascribe the extra variability in the new chronic disease cases that appear. And this is actually, this missing heritability is also due to the fact that there is so much complexity, that this complexity starts from the gene level or the cellular level, but extends into the macro scale of a disease phenotype at the individual level, at the neighborhood level, at the population level. And this complexity somehow needs to be comprehended, better understood, and gradually incorporated into our study design, into our methodological aspects. For example, one could consider a genetic disorder, PKU, phenylketonuria, that develops because of a mutation in a specific gene, the phenylalanine hydroxylase gene. So if a person has two copies of this mutated gene, then obviously they will not metabolize phenylalanine, a typical amino acid, after its ingestion with food. And then, in these cases, toxic products of metabolism of phenylalanine accumulate, and particularly in early life, this can be very serious and severe consequences because these toxic products of metabolism can damage the developing brain. Therefore, if you leave this genetic disorder untreated, then this can eventually evolve into intellectual disability, seizures, and behavioral problems. So, considering these genetic contributions to this phenotype, it would be interesting to learn that when this disease is actually tackled or treated with an environmental intervention, for example, given that we know this gene mutation to a particular mother and a newborn baby in particular, then a special low phenylalanine formula, which is an environmental intervention, could potentially reverse and not allow these toxic products of metabolism to accumulate and instigate the damage in the developing brain. So, despite the fact that we consider these genetic diseases, obviously we have to consider also how the environment could potentially interact with the genotype in order to dictate eventually the disease phenotype. So the human exposome is actually a life course framework that holistically characterizes all the non-genetic, basically all the environmental components of chronic disease, and not only characterizing them, but also integrates them, with the different layers of multiomics. And by doing this, we actually enter the new biology era, including the exposomic applications in many different interesting approaches, cases, models, and new applications. So in this graph, one can consider that a human being, actually internally, we have this internal exposome domain. So the human exposome is actually comprised of three major domains. The first one is the internal exposome domains. So this could actually relate to various intrinsic properties, age, our medical history, our omic profiling, for example, blood pressure, etc. And as an onion with the different onion layers, as you extend outside from the internal physiology and its characteristics, then we actually talk about the specific external exposome domain. So this comprises all the exposures, all the environmental exposures that a human being actually gets in contact with during their everyday life. So this can be classic environmental stressors like water contaminants, air contaminants, food contaminants, but the specific external exposome domain does not stop including those classic environmental stressors, but obviously includes also other behaviors or lifestyle choices, smoking, alcohol habits, cooking habits, physical activity, screen time exposure, anything that actually humans get in contact with during their everyday life, at home, at work, outdoors, and elsewhere. And the human exposome, as an onion layer, further extends outside the human being, and that is basically the general so-called external exposome domain. So this is the third and last exposome domain, which groups together different exposomic components such as built environment characteristics, various climatic components, healthcare access, education, socioeconomic status, green and blue spaces, you name it. Everything that is actually dictating eventually the kind of exposures that will reach, and the type of exposures and the level of exposures that eventually then will reach the human being with a specific external exposome domain, and then will stimulate an endogenous response within the internal exposome domain. So there are different definitions and different, I would say, articulations on how to basically holistically capture the human exposome, which is basically a totality of exposure from conception, from the moment a baby is conceived until the death of the adult. So just like we talked about already, the three different major exposome domains, and the fact that we have some more genetic disorders that are very clearly attributed to genes. So diseases like type 1 diabetes or the PKU we just talked about, or cystic fibrosis. And on the other end of the spectrum, you have major environmental, let's say, attributed morbidity and mortality burden of disease, such as various injuries, accidents, etc. And then in the middle, we have all these non-communicable diseases for which we should consider that an interplay of genes and environment is responsible for the majority of these new incident disease cases. So there have been some developments and some definitions of what the exposome is about, and there is another definition by Miller and Jones which talks about the human exposome as the cumulative measure of environmental influences and the associated biological responses throughout lifespan, including not only external exposures from environment, diet, behavior, but also endogenous responses. And some more recent exposome definitions based on the European Parliament's Human Exposome Research essay that actually describe the concepts of the exposome and the exposomics. So the definition, the exposomics is basically a new transdisciplinary field that is aimed to a comprehensive and discovery-based understanding of how the exposome influences biology and health. So this is a short version. You can also see the extended version. And there is also an exposomics definition for the general public, which is, in a short version, it's basically the comprehensive study of the effects of environmental factors on health. Simple as that. But despite that, we have not invested a lot as a society on better understanding how the environment eventually dictates, shapes disease process. one of the reasons why we haven't really invested on the human exposome, at least in analogy to the investment established for the Human Genome Project, is the fact that human exposome is complex. And it's complex because it's by nature very dynamic in time and space. There are some tools, exposomic tools, sensors, biomarkers, various multi-omics assays, and various geospatial and other remote sensing or satellite, for example, models of characterizing the environment, together with new biostatistical time and space varying algorithms, various machine learning algorithms basically, that are becoming more and more useful in this kind of investigations. So the so-called exposomics era, which is part of this new biology era, considers that genomics is basically as a function of gene expression more than simply a variation in DNA sequence. So if you bring together the exposures in the picture, so together with the exposome, we include these exposures, these external environmental exposures, to dictate and influence the transcription of the genome and the translation with the transcriptome, and how the RNA translation actually dictates the number and type of the different protein synthesis or modifications that take place in the human physiology, and even going more downstreams, how then the metabolome or the various metabolites, these short molecules that eventually are produced or not produced, depending on the functional processes that take place, would eventually the integration of these different layers with the help of multi-omics data and multi-omics data processing algorithms, eventually, if one would prospectively monitor this in time, we should be in a much better position to predict or prevent disease phenotype by classifying, let's say, the phenotype, the disease phenotype, to different risk stratified disease groups. For example, higher risk, middle risk, or low risk. And right now, as we speak, there is a huge momentum for the human exposome concept. There are different programs that are thriving in both sides of the Atlantic, both in the US and also in the European Union. For example, in the past, the HERE program of the US National Institute of Environmental Health Sciences, together with NIH, funded All of Us research program, which gathers health data from more than 1 million people living in the United States, and now they're considering of including also exposome data for these participants. In Europe, we have the European Human Exposome Network that comprised of 9 different exposome-focused projects, and much earlier the EUFP7, the Helix, Exposomics and HEALS project that actually generated the first evidence of the human exposome research in the European Union. And now the continuation of the EHEN is basically the IHEN, which is a global network of exposome scientists, has been actually recently formed, and this is the International Human Exposome Network. And similarly to this IHEN network, there is also the US Nexus Network program, which is an analogous initiative by the National Institute of Health funding to establish a similar network of exposome scientists in the United States and beyond. So I will briefly go to some major exposome domains. So as you can see here, there was a scoping review that we conducted in order to identify which are the major external, let's say, group of components that are mostly published in the literature, which are the specific external groups of exposome components, and which are the internal groups of components that are mostly reported and used in exposome research. So, for example, we found that the papers the publications, the original peer-reviewed research articles that included all three major exposome domains, the general external, the specific external, and the internal exposome domains, which were 37 of them. We had about an equal number of studies that focused only on the specific external and the internal, and we had very few studies exposome studies that talked about only the internal exposome domain. So these are some, let's say, typical groups of exposome that were most frequently or less frequently included in these publications. So for example, you see that very few publications talked about policies and programs or psychological and mental stress, whereas a big emphasis was given on the natural and built environment and information on exposure and information on socioeconomic status. Similarly, for the specific external group of components, we can see here that most of the studies included information about particulates in air and gaseous pollutants, very little emphasis on noise, soil contaminants, or water contaminants, as you can see here, or infectious agents. So you can see here by looking at this table that there have been some either gaps or emphasis of the exposome research as it tries to mainstream and move forward in the future. And briefly to go through the major exposomic tools that are applied in practice in addition to the structured questionnaires, surveys or interviews. As you can see here, half of the studies, 47% of the exposome published studies included structured questionnaires or interviews in place. But we also had biomarkers. 36% of these studies included biomarkers of exposure or biomarkers of effect. About 1 in 4 of these papers included some kind of omics platform in the paper. Very few of them incorporated interactions with genes, such as genomics. So this is obviously a critical gap that the exposomics need to fill in together with geneticists. Application of secondary data, as you can see here, sensors, not much, but obviously these are some emerging exposomic tools that find more and more use nowadays, together with remote sensing, satellite sensing, and geospatial exposome modeling. This is very hot topics right now. So the way we can analyze the data that emerged from this is actually very, very important. In analogy to the genome-wide association studies, we also have examples of publications that focus and apply these techniques. This one exposure with the outcome association by including multiple one at a time exposure that is included in these regression models. We don't have time to go through them. This is briefly just to mention this and how, where and when these biodynamic trajectories of genome and exposome interactions meet, and some applications of the human exposome to the urban exposome and how the urban health of populations can be better characterized, integrated, and understood in urban settings. So I would like to close basically by highlighting that the human exposome is here. The future of chronic disease prevention and control will be dependent at large to the growth of the human exposome methodological framework and its tools. And thank you very much for your attention.